Imagine a switch in the brain, a single point of control for the swirling chaos of anxiety, depression, and the struggle to connect with others. For years, scientists have understood that our deepest fears and emotional responses are rooted in a small almond-shaped structure deep within the brain known as the amygdala. But pinpointing the precise mechanism of malfunction has remained an elusive puzzle. Now, a groundbreaking discovery by a team of Spanish researchers has finally illuminated the core of this imbalance. The news that has electrified the neuroscience community centers on the amygdala, a region long recognized as the brain's alarm system. A group of scientists led by Juan Lerma at the Spanish National Research Council and the Miguel Hernández University in Elche has uncovered a specific neural circuit within this tiny structure that, when out of balance, seems to be a direct cause of severe emotional and social disorders. Their work suggests that we might be on the verge of a completely new way to approach mental health conditions that affect hundreds of millions globally.
For decades, we've known that the amygdala is where we process emotional memory and respond to threats. When it perceives danger, it signals a cascade of reactions, priming the body for fight or flight. In a healthy brain, this system activates when needed and then quickly returns to a calm state. But for those suffering from chronic anxiety or depression, this system can remain stuck on overdrive. The Spanish team didn't just observe this, they found a direct causal link, identifying a critical imbalance in the activity of specific neurons within this emotional command center. The crucial finding centers on a complex interplay between different types of nerve cells. The brain, at its most basic level, operates on a delicate equilibrium of excitation and inhibition. Think of it like a perfectly tuned engine. You need both the gas pedal, excitation, and the brake, inhibition, to function smoothly. The researchers zeroed in on a specific group of neurons in the amygdala that are associated with a gene called GRIC4.
This gene, GRIC4, is responsible for producing a type of glutamate receptor known as GluK4. Glutamate is one of the brain's major excitatory neurotransmitters. It's the gas pedal. The scientists discovered that in the brains of the mice they studied, there was an overexpression of this GRIC4 gene, which led to an excessive number of these GluK4 receptors. The result was a heightened level of excitation. The gas pedal was pressed down too hard, creating a state of hyperexcitability in these key neurons. The consequences for the mice were significant. These animals began to display behaviors strikingly similar to symptoms observed in human conditions like autism and schizophrenia, characterized by profound anxiety and a noticeable deficit in social interaction. They became withdrawn and highly agitated in novel or open environments, typical markers of intense anxiety in these models.
The genius of this study was not just identifying the problem, but also finding a way to correct it. The researchers were able to precisely intervene and restore the normal balance of activity in the basolateral part of the amygdala, an area central to emotional processing. They re-established proper communication between these hyperactive neurons and the inhibitory neurons in the central lateral area. And here's the truly astonishing part. Simply normalizing this one specific neural circuit was enough to completely reverse the pathological behaviors. The anxiety dissipated, and the mice's social skills improved dramatically. The aggressive or withdrawn behaviors vanished. It was a targeted correction that delivered a profound transformation, proving that this specific neural imbalance was the root cause of the observed symptoms. This isn't just about tweaking a few neurons. It's about understanding the core machinery of fear. The amygdala is not a singular entity, but a complex network of interconnected subregions. The BLA, or basolateral amygdala, is crucial because it acts as the primary sensory input hub, taking information from the outside world and sending it deeper into the brain. It is here that emotional significance is assigned to stimuli. If the BLA is overactive, everything can feel like a threat.
The study highlights a concept known as the excitation-inhibition balance, or EI balance, which is fundamental to how the brain operates. When the EI balance is disrupted, typically with too much excitation, the brain's networks become disorganized, leading to a host of disorders. In this specific case, the GRIC4 gene disruption tilted the balance towards excitation, essentially locking the amygdala in a state of high alert.
The researchers used sophisticated electrophysiological methods and behavioral tests to monitor the mice. They observed how the animals reacted to open spaces, which typically trigger fear and avoidance in anxious rodents, and how they interacted with their peers. After the genetic correction, the mice that had previously been highly anxious now explored the open environments with ease and engaged actively with other mice. What makes this discovery particularly powerful is its universality. The Spanish team even performed the same correction on ordinary mice that naturally displayed anxious behavior and observed the same positive results. This suggests the mechanism they uncovered is a common pathway for anxiety, regardless of whether it's genetically induced or a natural predisposition.
However, the findings also came with important caveats. While anxiety and social deficits were erased, some cognitive issues remained. For example, the mice still struggled with certain tasks involving object recognition. This observation is vital because it confirms that mental disorders are multifaceted and involve multiple brain regions. It underscores the critical role of other areas, such as the hippocampus, which is primarily responsible for memory and spatial navigation, in processing and integrating cognitive information. The hippocampus and amygdala are constantly communicating. The amygdala provides the emotional context, while the hippocampus provides the memory and spatial map. If the amygdala is too active, it can hijack the hippocampus. But if the hippocampus itself is malfunctioning, simply fixing the amygdala won't solve all cognitive issues. This Spanish research on the GRIC4-related imbalance offers a powerful new direction for treatment development. Historically, medications for anxiety and depression, such as selective serotonin reuptake inhibitors, or SSRIs, have worked by modulating the overall level of neurotransmitters in the brain. They are often effective, but they are also broad-spectrum, meaning they affect many different parts of the brain, leading to side effects.
The approach demonstrated by the Lerma team is entirely different. It represents a precise, targeted correction of a specific circuit. Instead of adjusting the entire engine, they are fixing one crucial broken part. This kind of targeted neuromodulation holds the promise of therapies that could be far more effective and have fewer unwanted side effects. The brain's complexity is daunting. Scientists are still grappling with how the amygdala interacts with the prefrontal cortex, the seat of executive function and higher-level thought. The prefrontal cortex is often considered the brain's rational center, tasked with calming down the amygdala when there's no real danger. In many cases of chronic anxiety, the communication between the prefrontal cortex and the amygdala is disrupted, with the alarm center overwhelming the rational center. The discovery that an imbalance in GLUK4 receptors can cause such severe behavioral symptoms sheds new light on the interplay of genes and mental health. While genetic factors are known to play a role in disorders like schizophrenia and autism, understanding the precise pathways through which these genes exert their influence has been challenging. This research provides a clear roadmap, linking a specific genetic mutation to a measurable neural imbalance and observable behaviors. The implications for human health are profound. If we can replicate this targeted correction in people, we might be able to treat the underlying cause of anxiety and social deficits rather than just managing the symptoms. While my studies are a vital first step, translating these findings to humans presents significant challenges. The mouse brain, while structurally similar to the human brain in many respects, is far simpler. Techniques used in this study, such as direct genetic modification, are not currently feasible for therapeutic use in humans.
However, this research is not merely theoretical. It opens the door to new drug development.
Knowing that GLUK4 receptors and the associated EI balance in the amygdala are critical to emotional regulation allows researchers to search for pharmaceutical compounds that specifically target these receptors. If a drug can selectively reduce the activity of these hyperexcitable neurons, it could offer a novel therapeutic avenue for those struggling with severe anxiety and social withdrawal. The concept of precise neural balance is becoming a dominant theme in modern neuroscience. The work of Lerma's team underscores that the brain's harmony relies on the precise calibration of its circuits. When that calibration is off, even in a specific localized area like the amygdala, the downstream effects can be devastating, impacting everything from our deepest emotions to our ability to interact with the world around us. This discovery is a major stride forward in our understanding of the brain's emotional architecture. It provides a biological blueprint for anxiety and social disorders, moving the discussion away from purely psychological perspectives and towards a concrete biological basis. The potential for precision medicine in mental health has never been closer. We are moving towards a future where therapeutic interventions are tailored to the specific neural circuits that are out of balance, promising a more effective and less disruptive path to recovery for those living with these conditions. The journey from the lab bench to clinical application is long, but this breakthrough offers a powerful beacon of hope.